Da ønsker jeg velkommen til denne presentasjonen av en del av den forskningsaktiviteten som vi holder på med ved Institutt for biovitenskap når det gjelder appetitt og fôrinntak med fokus på laks. Det min oppgave å gi en veldig kort oversikt over en del av det vi holder på med, og så, som ble sagt her, så vil det også være 2 presentasjoner av henholdsvis Tharmini og Floriana som presenterer doktoratsarbeidet til Tharmini og et prosjekt som Floriana jobber med. Der var han. Ja, så i de prosjektene som vi presenterer nå, da blir arbeid utført sammen med gruppa til Sigurd Handeland. Så sammen så har vi en del forskere, Floriana, Anna og Pablo, og i tillegg en del stipendiater som jobber mot feltet appetittregulering. Det Tharmini, Linn, Sissel, Hanne Joren og Endre. De 2 siste i industri nærings-PhD, og så har vi også teknikere i gruppa. Og Sigurd har også en stipendiat, Enrique, som har levert sin PhD og presenterte vel i et tidligere Impact-forum. Vi jobber med mange fiskeslag. Vi ser noen av de her. Kveite, klovnefisk, leppefisk, kobia, mens det vi skal fokusere på her laks. Så appetittregulering hos laks. Nå har vi jo, i hvert fall de fleste av oss, akkurat spist lunsj, og da det lett å relatere seg til det som går på appetitt. Veldig forenklet så kan en si at appetittregulering det en syklus mellom 2 hovedstadier. Det en har behov for å ete, altså en får en sultfølelse som blir sterk nok til at en begynner å ete, og når en da etter en stund får mettelsesfølelse, så vil de fleste da stoppe å ete. Det kaller vi på engelsk satiation. Så går det også vanligvis en del tid mellom måltid, der en føler seg mett, før igjen sultfølelsen bygger seg opp. Det her noe som vi alle kjenner godt til, altså det med sult og metthet. Så det 2 viktige begrep, altså hunger og satiation, sult og metthet. Så det vi forsøker å forstå mer av det hvordan blir det her regulert og oppfattet av fisk. Når det gjelder, skal vi se, flytte i menylinjen her, så kommer vi til veien. Når det gjelder, dersom en ser på den nederste her, så det sult, altså hunger. Framfor et måltid så vil de fleste føle seg sulten. Og mens en spiser så vil den sultfølelsen forsvinne, og så vil en føle seg mett på slutten. Så går det en viss tid, og så bygger det seg opp en ny sultfølelse, og så det da, hvis mat tilgjengelig, så vil en da kunne innta et nytt måltid, og så vil en føle seg mett der også. Så det vi forsøker å forstå, det altså signalet som starter et fôrinntak dersom mat tilgjengelig. Og vi ønsker også å forstå signal i fisken som gjør at fisken føler seg mett og stopper å spise. Så vi har fokus både på start av måltid og på avslutning av i. Og spesielt det her har veldig stor innvirkning mot oppdrett, fordi at hvis en bommer på det her og fortsetter å fôre etter at fisken er mett, så vil en da kunne kaste bort mye fôr og få et stort fôrspill. Så spørsmålet er også, dersom en har påfølgende måltid, hvor sulten er fisken, og hvor mye vil fisken spise ved det neste måltid. Altså hvor mye blir han påvirket av det forrige måltidet. Når det gjelder reguleringen av det her med sult og metthet, så er det prosesser som skjer i hjernen. Hjernen har kontrollsenter som styrer appetitt, og det gir seg utslag i fôringsatferd. Så fôringsatferden er styrt i et område i hypotalamus, regner vi med, og det gjør at fisken, dersom det er mat tilgjengelig, så vil den kunne innta den maten. Skal vi se denne menylinjen, den kommer. Ja, som jeg sa, så blir appetitten regulert i hjernen. Det kaller vi den sentrale appetittkontrollen. Men i tillegg så får også hjernen informasjon fra andre deler av kroppen. Det er fordøyelsessystemet, mage, tarmtraktus, gir mange ulike signal om magefylling, tarmfylling, som er viktig for avgjørelsen om hvorvidt fisken vil innta mer mat eller føle seg mett. Og det er også signal fra andre deler av kroppen, muskel fra lever, som gir tilbakemelding, det vi kaller perifer kontroll av appetitt, til den sentrale delen av appetittkontroll i hjernen. Basert på den korte introduksjonen her, så vil nå Tharmini gi litt mer detaljert informasjon om de studiene hun har utført for å få mer kunnskap om de mekanismene som styrer appetitt hos laks. Så da gir jeg ordet til Tharmini.
Hello everyone, I'm Tharmini Kalananthan, and my main supervisor is Professor Eva Ronestad, and co-supervisors are Professor Sigurd Handeland and researcher Floriana Lai. I am going to present on the title Atlantic Salmon Post-Smolt, Basic Mechanisms for Control of Appetite and Feed Intake. So in this presentation, I will present a brief introduction and the methodological approach, results, and finally the summary. We all know there are external factors like environmental factors, physical factors, and social behavioral factors affect the appetite and feed intake in Atlantic salmon. But our main focus in this study to see how the internal factors affect the appetite and feed intake in Atlantic salmon. To understand more on the internal control, it is better to have a look in the mammalian model, how this is controlled by internal factors. So in this figure, we see, as previously Professor Ronestad mentioned, the brain is the central control of appetite, and especially in the brain, the hypothalamus is the region where which is the feeding center for appetite regulation. In hypothalamus, there are two regions, arcuate nucleus and paraventricular nucleus. In arcuate nucleus, we see different types of nerves, which produces the signaling molecules like AGRP and POMC or CART AGRP, NPY, or POMC, and CART. And these interact with the other nerves in the paraventricular nucleus and transit the signaling pathway. So all these nerves, including the signaling molecules and the receptors from these nerves, are called the Melanocotine system. And depending on the nutritional status of the animal, the peripheral signals like insulin, leptin, and CCK from the peripheral region, the signals activate the POMC when the animal is in the fed state or satiety state. These signals activate the POMC or CART nerves, and this will activate the Mc4 receptor. The production from these nerves, alpha MSH, binds with this receptor and inhibit the food intake. On the other hand, when the animal is in the fasting or hunger state, the ghrelin, which is the product from stomach, activate the AGRP or NPY nerves, and this will produce these AGRP or NPY signaling molecules, and this will inhibit the action of Mc4 receptor and activate feed intake. So if we look at the mammalian, if we look at the Atlantic salmon gut-brain axis appetite control, keeping this mammalian model in mind, we have the central candidate signaling elements as we saw here, the same NPY, AGRP, CART, and POMC from the brain. And from the peripheral system, like digestive system, stomach, and mid-gut, from stomach, ghrelin and MBOT, and gut, CCK, PYY, and GCG. In the previous studies, the whole brain was utilized for analysis of these signaling molecules. But we have seen in other fish species, there are studies showing the dissected regions of the brain for this analysis. So we have dissected the brain in this current study into 8 brain regions like olfactory bulb, telencephalon, midbrain, cerebellum, hypothalamus, pituitary, sulcus vasculosus, and brainstem. So if we to see how all these genes, these signaling molecules are observed in these regions, we have carried out. We can see the objectives of this study. So we see the first objective to establish and validate the assays and protocol for dissection of brain regions for qPCR analysis of these candidate appetite genes. And the second one, to study the brain distribution of candidate appetite genes in 8 brain regions and the impact of short-term fasting in these genes. And the third one, to study the response of long-term fasting on candidate appetite genes in gut-brain axis. So this flow chart shows how the brain samples and the gut samples were collected in the all experiments, and the brain samples were dissected, like as I mentioned before, 8 brain regions, and this is the protocol flow chart. Parallelly, we have seen in other species there were multiple signaling molecules types existed. So we have searched in the genome databases, because Atlantic salmon also went through the whole genome duplication events. So we searched in the genome databases and found many multiple signaling molecules. So parallelly we have done some assays validation, and we continued with the qPCR at last for all these identified genes. So in the following chart, we will see how from the genome database research, we found For each of these mammalian signaling molecules, we found from the central Melanocotine system 2 AGRPs and 3 NPYs and 10 CART and 3 POMCs and 4 Mc4 receptors. From the peripheral regions in stomach, we found 2 ghrelin and 1 MBOT, and from the gut, nearly 9 CCKs and 4 PYYs and 2 GCGs. These are still in the process of assay development, and we call these with the following names like AGRP1 and 2, NPYA1A2B, and CARD as shown here 1A24. But we could only manage to develop 7 assays to cover all these 10 cards. So these 2 were really difficult to differentiate. So we have 1 assay for these 2 types of card. So to see all these signaling molecules, how they are available or observed in the brain regions, we have conducted an experiment to see the brain distribution and the impact of 4 days of fasting. So here we have 2 groups of Atlantic salmon post-moles. One group was fed, normally fed for 4 days, and the other one was fasted for 4 days. And we expected to have, as we have seen in the mammalian model, NPY as a hunger signal inducing the food intake. We expected to see in the hypothalamus, because hypothalamus was the feeding center in the mammalian model, so high expression in the fasted group. But we have seen 3 types of NPY in the different patterns among all these regions, the level, but NPY-A1 shows the higher level, the total higher level, and this is found in mostly in these regions, whereas the NPY-A2 was highly found in the hypothalamic region. But we see only one, the NPY in the midbrain region, statistically different in the in the fasted group, but there is a trend of higher level in the hypothalamus fasted group. So this is quite similar action as in the mammalian model. If we look at the CART signaling molecules, as I mentioned before, we have analyzed 7 assays, and CART2B is the highest one among all the 7. And we see the CART only in the CART2B. There are statistical difference between the fed and the fasted group. But the fasted group shows the higher level here. This is contradicting with the mammalian model, because a CART in mammalian model is a satiety signal. This will inhibit the feeding. But here we see this looks like inducing the feeding. And in the other signaling molecules, we have seen all these 4 types of melanocotine receptors. They have a quite similar pattern, like telencephalon and hypothalamus have higher level of these receptors. But here also we haven't seen any difference between the fed and the fasted group. Among the POMC types, all these 3 types show higher level in the pituitary, hypothalamus, and sulcus vasculosus. These 3 regions, they have a similar trend. But in between the fed and the fasted group, in the hypothalamus, we haven't seen any statistical difference, but we see here a smaller decline in the fasted group, and there are individual variations here. But according to the mammalian model, this POMC also satiety signals, so we expect to have the higher level in the fed group and low level in the fasted group. Among these 2 AGRP groups, we see the AGRP1 is highly available in the hypothalamus, and AGRP2 is higher in the telencephalon. Even in this case, also we haven't seen any statistical difference between the fed and the fasted group, but we see a slight higher level in the fasted group for AGRP1. As we have seen in the mammalian model, AGRP1 is supposed to be the hunger signal inducing the feeding. So if then we have another small experiment where we have only analyzed the hypothalamic region. This is the 3 days of fasting, and we analyzed only few genes like AGRP1, AGRP2, POMCA1, POMC2, POMCA2, and POMCB. Here we see, like in the mammalian model, the fed group has higher level, fasted group has a higher level of AGRP1 in the hypothalamus. That means this is inducing the food intake. Among the POMCs, we see a trend of decline in the statistical trend of decline in the fasted group. So here, this is also one of the satiety signal according to the mammalian model. So in the long-term fasting experiment, we have also analyzed the 4 to 6 weeks of long-term fasting experiment. Only the hypothalamic region was analyzed. And here, again, we see the AGRP, the higher level of expression for the fasted group, both in the 4 weeks of fasting and also in the 6 weeks of fasting. And also this black and white graph shows these fed groups don't have any difference between them. So here, this is another clear hunger signal is observed in the AGRP1. And we selected only AGRP1 for this analysis because from the brain distribution analysis, we have seen the higher expression level only for the AGRP1 in hypothalamus. Similarly, for the other signaling molecules types, we selected NPY-A1 and A2 from the hypothalamus, and we see NPY-A1 at 4 weeks of fasting had a higher level of expression in the fasted group. So this is also here, but there was no any difference between the fed and the fasted group for the NPY-A2. So out of these three, supposed to be the hunger signal in the mammalian model, we see AGRP1 and NPY-A1 showing the hunger signal or inducing the feed intake in Atlantic salmon. If we look at the CART genes, we have selected 4 CART signaling type. And here I show 3 of them because these 4 were higher expression level found in the hypothalamus. And again, as we saw in the previous short-term fasting experiment, the higher level was found in the CART2B. And here we see the CART2A also shows higher level of expression in the hypothalamus for the 4 weeks of fasted group. But there were no any difference between fed and the fasted for the other 2 CART types. So out of 4, we see these 2 CARTs showing an opposite trend to the mammalian model. Here, this seems to be inducing feeding or is contradicting results to the mammalian model. If we look at the, this one also one of the other CART, as I mentioned before. If we look at the POMCs, we selected only POMC A1 and A2 for this analysis, and we see both showing the similar trend in the fasted group, only at the 6 weeks of fasting. Here, this POMC also shows a higher expression level in the fasted group, opposite to the mammalian model trend. Then we see next the Mc4 receptors. We selected only 2 receptors which had the high level of expression in the hypothalamus. out of these two, MC4A2 and B1, we see MC4A2 shows higher level of expression in the both group and statistically different in the 6 weeks of fasting group. So this is not clear why the fasted group show the higher level of expression, because MC4 is important for the satiety signal in mammalian model, but it's not so clear whether this will, the level of expression for these genes will change with the feeding of fed or hunger situations. And in the another long-term fasting study, we in the same long-term fasting study, we see the stomach genes out of all those 3 selected signaling molecules, as I showed in the chart. Ghrelin 1 shows the decline in the fasted group of 6 weeks, whereas the other 2 doesn't have any difference between the fed and the fasted group in both time points. But we have seen in other literatures or other studies also supporting results, the 2 weeks fasting has declined the ghrelin 1 expression level in stomach. But actually this ghrelin also hunger signal in mammalian model. But it could be because of the long-term fasting, it could have adapted to the satiety signal here and may be available as a hunger signal when the food is available in the vicinity. So this is what we have observed so far for these all genes which we have analyzed in this chart. Here, all these marked genes showed higher expression level for the fasting group, increased with the fasting, and there was only one gene, ghrelin 1, declined with the long-term fasting. So if we compare these genes with the mammalian model, as I mentioned before, AGRP and NPY are supposed to be the hunger signal in the mammalian model. And here in our studies, AGRP1 shows both in the short-term and the long-term fasting hunger signal. And in NPY, we see NPY-A1 in the long-term fasting as the hunger signal, and NPY-A2, there was a trend of hunger signal for the short-term fasting. But CART, POMC-A, these 2 are satiety signal in the mammalian model. Here, this will inhibit the food intake. But in this study, it has given the opposite trend or opposite action. So this is still not clear, maybe because we have seen in the literatures the CART and POMC, they have the other functions also, like stress response or different many other functions. So maybe because of the fasting induced stress, this might have caused a higher expression level. So this is still a question. And MC4, we have seen it's important for the satiety in mammalian model, but here we have, there was no any difference in the short-term fasting experiment, but we see a higher level of fasted group in the long-term fasting for the MC4A2. Among the ghrelin, the stomach ghrelin is the hunger signal in the mammalian model, and we found ghrelin 1 as adapted satiety signal in the long-term fasting in Atlantic salmon. So our overall goal to find a good stable appetite gene that could induce the food intake, and it seems like from our study, both short-term and long-term, AGRP1 could be a good candidate, but still it needs to be further confirmed. Thank you.
Ok, så da har Tharmini gitt en litt mer detaljert innføring i den sentrale delen av appetittkontroll. Det er mange ord her, og mange signalmolekyler. Jeg kan bare si det at hun snakket mye om kart. Hos mennesker så betyr det altså kokain- og amfetaminregulert transkript. Det er en appetittregulerende neuropeptid som en ser blir påvirket hos folk som tar amfetamin, og det er altså en av grunnene til at matinntaket blir mindre. Og vi finner det samme signalmolekylet hos laks, og som sagt så er historien der mye mer komplisert, så Tharmini har brukt mye tid å nøste på det. Så da har det fått en innføring i den sentrale appetittkontrollen. Da går vi tar ett steg tilbake og ser litt laksen utenifra, fordi at appetittkontroll har jo med fôrinntak og fôringsatferd å gjøre. Og da er det samspillet mellom tilgjengelig mat og fôrinntak, og fôrspill som er fokus for den neste presentasjonen. Og det er et prosjekt som vi har gående nå, som heter No Food to Waste. Det er Floriana som vil presentere. Hun er forsker i forskningsgruppa. Så da gir jeg ordet til Floriana.
I'm going to give an overview of one project that we have in the group, No Food to Waste. It's a 3 years project started at the end of 2020, and it's an innovation project funded by the Norwegian Research Council. Being an innovation project, it aims, or like it builds on innovative ideas and development of new research technology that can be beneficial for the industries and society. The No Food to Waste project, as I'm going to introduce it, is going to focus on optimizing the feeding in the fish farms, feeding control in the fish farms. The project is a collaboration between industries as Lingalax and KGI, where Lingalax is the owner of the project, and a collaboration of national R&D partner like us, the group team from University of Bergen, so Professor Ronnestad, myself, Professor Handeland, and Pablo Balseiro. The project also includes a collaboration with the international partners from University of Stirling and the National Research Institute of Aquaculture in Japan. For this innovation project, we included a reference group, a group of Norwegian farmers as Bremnes, Eide, Leroy, and Greg Seafood. This group has been established to ensure a good dialogue with the Norwegian fish farms, but also to support the project with knowledge and experience, of course. So the No Food to Waste. So as the title already tells, the key of the project is to decrease the waste of feed. Feeding is one of the most important and expensive activity for a farmer, and of course optimizing the feeding is beneficial both for the fish growth and the welfare, but also for the industry economical cost. So it's quite common nowadays to feed the fish by subjectical observation of fish behavior, at least in the sea cages, through the use of underwater cameras. The feeder observe the swimming pattern and feeding behavior of this fish, and provides the feed depending on their location inside the cage. It's also quite common to provide like a good growth to overfeed the fish, which can lead to a high feed conversion rate, but also to a waste of feed inside the cage and in the surrounding environment, being therefore not like eco-friendly. So, and of course, in a case of adjustment in the feeding protocol, the farmers don't want to end up in a situation where underfeeding can affect the growth of the fish, and therefore even increase more the feeding conversion rate. So the No Food Waste project therefore aims to improve this feed utilization, fish welfare, and maximize the fish feed intake and growth, but at the same time minimize the feed emissions to the environment. So like building a more sustainable fish farming, but as well reducing the feed cost for the industries, which can be up to 2 million circa. So to achieve the main objective of the project, we are going to use two main tools. One is the use of innovative technology that has been developing by the KGI. It's EcoSounder that register the fish behavior, both like vertical and horizontal movement of the fish inside the cage. And I'm going to talk about that in the next slides. And together with that, we also use another tool, which is based on research on basic or basal physiological knowledge on the mechanisms that regulate appetite, feed intake, feeding behavior in relation to the size, the growth of the fish, and under different environmental conditions and seasons during the year. Therefore, we aim to improve the feed utilization and fish welfare, maximize the fish feed intake and growth, minimize the feed emission to the surrounding environment, so having a more sustainable fish farm, and also reduce the high production cost for the farm. The project is built under like 4 sub-goals. And the first sub-goal is an experiment, a large-scale experiment, where we want to map or actually we want to follow the salmon production during an entire cycle, and we want to map the appetite, feeding intake, feed utilization, growth, also under different environmental parameters and changes in these parameters, as temperature and oxygen during the different seasons and during 1 year. This large-scale study started this year in March in one of the locations of Lingalax called Bergadalen. In this experiment, we are using the innovative technology that I mentioned before, developed by KGI. The technology is based, as I was mentioning before, on the observation and of the fish behavior inside the cage. This technology is works through like hydroacoustic technology through the presence of two sensors inside the cage, one at the surface and one at the bottom. And when the fish goes to a specific area inside of the cage, that's we can call it like the feeding area, is where then a signal is sent to a program that tells the feeder that is the right time to feed. So I'm going to show you a quickly movie about it. So right now, yes, so we can observe the swimming of the fish, and as I said before, there are two sensors inside the cage that's managed to locate exactly the same position of the fish inside the cage and send this behavioral data to a softwares and suggest then the feeder when starts the feeding. As you see, when the fish goes towards the surface, is a sign that the fish is hungry, and then as soon as the behavior partner changes and the fish goes more towards to the bottom, is a suggestion that the fish is full. So from this, so this is like a machine under development, and until now we have been collecting data daily, and as you can see here at the top is like a representation of the echogram and that shows like the density of the fish along the water column. So brighter is the light, higher is the density of the fish, and to the right you can see like the depth inside the fish tank. And you can see how the behavior of the fish changes along the water column. And in particular, if you have a look at the bottom, when the feeding inside the cages starts, is when also correlates with a different, like a change in behavior of the fish that moves towards the surface. While it decreases and goes more towards to the bottom when, yes, the fish is full, and therefore then the feeding should decrease as well. So with this data, we can have a bigger overview of the fish feeding behavior and appetite. Here is a representation of a 1 week overview, in where we can see here at the top the daily feed provided to the fish, and also how like the behavioral pattern of the fish changes day by day. So it's like, so the fish goes more towards the surface during the night, then goes towards the bottom during the day, and of course that is also correlated with the feeding related to the feeding behavior. And here is also interesting to see how, for instance, in the night we see like two, let's say, clouds, and then it's a sign of different behaviors within the same group, and probably here it was like some changes in the environment. So if I remember correctly, like a strong current in the water, which it's here represented by a different behavior pattern from these two different groups. So this program is under development, and is a program that is based on many algorithms, and is like under like a learning process. But also is a good support for the feeders to understand when it's better to start feeding the fish. And in the very last week is when actually this program started Anbefalinger. It's to suggesting to give like a proper support to the feeders on when like starting the feeding inside the cages. So we so we at the same time we together with the KGI data, these experiments also includes a daily profile of environmental temperatures, here represented by plot date graphs, and you can see from like May to until a few days ago how, for instance, the temperature in the water has of course changed due to the different seasons as oxygen and salinity. And together with that, we are also collecting some biometry data to follow up the growth and the performance of these fish. We have 6 main cages, and we have been collecting biometry data since March 2021, so this year. And we will continue with this collection of data until March, April 2022. This data shows how the fish are having a good growth, a good performance, but also just by chance, we also got interested, or like we have a small trial within the trial where within the population that we have 2 small groups. So we have 3 cages coming from a flow-through system, while 3 cages coming from the RAS system. And it seems actually there are some slightly differences in performance, but overall the performance of the fish are very good. So with all this data, so the data, we want to combine the data from KGI, so the behavioral data, feeding data, together with the environmental data and biometry data to study to study the correlation that there is between the appetite, feeding intake, and feeding behavior under different environmental condition within the different season during the year. In this way, we can start developing and models on how to optimize the feeding inside the sea cages and the fish farms. Therefore, meanwhile, this big scale study is going. We are starting also a small scale study at the University of Bergen. In this experiment, we want to have a deeper overview of the mechanisms that regulate appetite, digestion, and gut transit, as has been shown quite extensively by Tharmini. But in particular, in this experiment, we want to see how these mechanisms can be affected by temperature and oxygen levels, and therefore how this can affect the growth and the fitness of the fish. In this small scale experiment, we are going to have two small trials, one that will be focused on only temperature and one focused only on oxygen. So the idea is to have, we're going to rear fish at different either temperature condition or oxygen condition, and then follow up the mechanism that, as I said, control the gut-brain axis towards along 2 months trial. Therefore, in this trial, we are going to analyze or collect brain tissues, different 3 sections from the gastrointestinal tract, gut content, and also plasma. Therefore, in this experiment, we will come back to the hormones and peptides that belong either into the peripheral control group or central group, and control the food intake appetite in the fish. The last experiment of the project is to collect the information and knowledge acquired from the previous 2 tests, and validate the models on the feeding protocols into production cycle. One is a small scale experiment, and the other one will be more like in a large scale experiment. These experiments are going to be held in Matter at the IMR facility, and are planned to start by the end of 2022. So as you can see, we are still at the really beginning of the experiment, but of course, then being like innovation projects is also our main objective, to share our acquired knowledge on this modeling, share our results to breeders, farmers, and then of course authorities and general public. Yes, that was all. Thank you.